Welcome to this colloquium. It's the last before the summer, and I will just tell you in anticipation, we already organized one colloquium of the next academic year for October 29, Wednesday as usual, which will be devoted to very interesting experimental activity in quantum optics that holds the promise of telling us something about the quantum nature of gravity. Please.
So today's speaker will be Dario Valenzano, and it's really a great pleasure. Sometimes I introduce colleague with "it's a great pleasure," but it's kind of a formal thing. But in this case, it's really amazing having you here, because I could see you at the beginning of your studies during your PhD, because you have to know that Dario studied biology here, University of Pisa, and the Scuola Normale, and actually got the PhD from us, the PhD in neuroscience of the Scuola. And since then, because of his work here together with Professor Celerino, he managed to work on a very important subject on aging, and in particular introducing a new model, a new animal model for aging that has such a very useful characteristics that I'm sure we will learn from Dario's talk. And he went to Stanford for a PhD, then research associate, then in 2013 he moved back to Europe, in particular to Germany, to become group leader at the Max Planck Institute. And after that, since I think 2021, he moved to the former East Germany, to Jena, where he became a senior group leader, actually director of this Leibniz Institute, the Fritz Lipmann Institute in Jena, and is also, of course, full professor at the University of Jena. So I really look forward to your talk, and the floor is yours, and thank you for coming.
Thank you so much for the kind introduction. It's really a thank you, Tommaso, for the introduction. It's my pleasure really to be here today. It's really my honor. I can't believe I'm in Sala degli Stemmi presenting to you the work that we've done over the past few years, and really that started here. Thanks to Sandro, but also thanks to the whole Scuola that really gave us and me the opportunity to start this ambitious project a long time ago, where we really didn't have tools. We really had rough ideas about how to go by. And then the way things have unfolded, it's quite remarkable. And I'm really excited to share some of the work that we have, and others have done. And I would like, though, before getting there, to spend a few minutes introducing to you the field of studies that we work on and we focus on. So we are, broadly speaking, interested in the biology of aging, which is a quite abused term and quite confusing term and field as well. What we're talking about here is really what happened when you transition from a healthy adult individual into like a less healthy, frail individual. Of course, there is time that goes by, but there are a lot of changes that occur through this time that are relevant to biologists. And you may wonder, why are we even thinking about the science of aging? Why do we need a biology of aging? Isn't, after all, just wear and tear? Isn't just like accumulation of damage that we are talking about? Isn't it that any object, from the floor in this room to the facade of this building to our, computers, don't they just decay over time because of physical reasons and not because of biological reasons? Why are we even indulging into talking about the biology of aging? So what's the difference between how inanimate objects age and how animate objects, organisms, age? This is a fundamental point, and there is a fundamental difference in the way in which, and in the ways in which organisms age compared to inanimate objects. non-living organisms. First of all, organisms build up themselves. So they undergo a process which is called autopoiesis. So here you can see how a Drosophila single-cell embryo developed into a multicellular organism, and you have body patterning that emerges through time in a very orderly, in a very structured, time-wise and space-wide manner. So organisms build themselves up from ground zero, interacting with the environment, unlike inanimate objects that are built by someone else, by a builder, and then they are let decay on their own. So organisms really like build themselves up, and here is a salamander actually developing from, again, like a single cell, from a single cell, and here you have, of course, a plant seeds that it's developing into like a plantula, and then will develop into an adult plant. So organisms build themselves. This is a fundamental property that distinguishes, life from non-life, for example. Another key feature of organisms that demands our attention when we're thinking about the biology of aging as opposed to aging in general terms is that organisms not only build themselves, but also maintain themselves. So there are extremely sophisticated processes of actually damage avoidance. You can think about them as defense mechanisms, if you wish, if you allow me this term. So for example, in humans, but also in other organisms, integument and pigments can actually shield, can protect deep tissues from DNA photo damage. So absorbing certain wavelengths can prevent damage to actually DNA that might actually be causing trouble and disease. So this is the first way in which actually protecting, shielding yourself is a way to sort of maintain homeostasis and prevent damage-induced decay over time. This is another example. This is a clutch of frog eggs, and what they are characterized with is that they have a lot of mucus around them. In this mucus, in this substance, there is a lot of antifungal, antimicrobial compounds that shield again and protect the developing organism from external insults, in this case, pathogens. So this is a way to avoid prevent the damage that might incur from external insults. There are other ways in which organisms maintain homeostasis, which is by recognizing the damage has occurred, and then mounting a response to deal with the damage. Okay, so these are processes that require these two steps: recognition, identification of the damage, and response. So, for example, DNA, in cells. So there are mechanisms that recognize different classes of DNA damage, and then they recruit mechanisms to actually fix that damage, right? So there are extremely sophisticated molecular machines that are able to cope with damage, to signal there is a damage, and to recruit the machinery that repairs that. I won't go too much in detail. Same applies for proteins. Proteins here, rather than being fixed, actually are disposed of. So misfolded protein, damaged proteins, damaged peptides are flagged, are recognized, are flagged with particular type of moieties, and they are sent to the garbage disposer, the proteasome, for example, that actually grinds them down through this kind of channel and then provides the substrate for new proteins to be built up. So there is a mechanism of damage recognition at a level of multiple macromolecules, DNA, proteins, but also at the level of organelles, at the level of cells, at the level of tissue, at the level of organs. So I won't go into all the layers of biological complexity, but just to say that damage recognition and response is actually a fundamental process. And then there is actually even, next level response, which is regeneration. So once damage has been recognized, there is actually a way to actually fix it by actually regrowing the damaged tissue, by eliminating the damaged tissue and replacing with new tissue. So this is a classical textbook example of a salamander. This is an axolotl limb. So this is just like a picture of a hand of an axolotl, which is like a neotenic salamander from Mexico. It's an extremely useful model, laboratory model. It's been like this limb has been cut So kind of gruesome intervention, but don't worry, the limb will regrow over time. So there is a regrowth, perfect regrowth of this limb from the axolotl. They are adapted to regrow themselves. So damage can be not just recognized, but also fixed, and very complex organs can be rebuilt in several organisms. The same applies actually to us. Even now, our gastrointestinal tract, our intestinal epithelium regenerates itself on a 3-4 day, 5 days turnaround. So we completely renew our intestinal epithelium at different speed, at different efficiency. So we also do that in some organs. Okay, so what I told you is that defense, response, and recognition of the damage is really our key factors that contribute to homeostasis, to maintaining homeostasis. So if you might think, if you may be drawn to think that, thermodynamic, from an entropy standpoint, organisms should actually decay over time. Well, think that actually biological systems, individuals, if you wish, are actually open systems that exchange energy with the environment. So actually that really doesn't apply much to organisms. I'll show you just now how we're good at dealing with damage, repair, etc., etc. So aging is actually oftentimes recognized as the time-dependent failure in each of these processes of defense, shielding, processes of response, processes of recognition. So this is what we study. We study what goes wrong with time and what causes these changes that impact these kind of like safeguard mechanisms that together contribute to homeostasis, so that this kind of like age-dependent decline in the performance in each of those mechanisms might actually lead to organismal aging. Maybe, there is more to this, of course, and I don't have the time to go into that in depth. And some of you in the room are really experts in several of these aspects. So, this better than I do, but actually, sometimes it's actually overreaching performance in any of those that actually might also cause trouble. So if you actually do too much of defense, too much of response, too much of recognition, if you measure amplitude, that can actually contribute negatively to homeostatic responses. Okay, so this was my first part. The second part I want to make is about, other counterintuitive turning point in the history of this field of investigation, molecular biology of aging in particular. In 1993, Cynthia Kenyon from UCSF, University of California, San Francisco, published a paper. She was a junior group leader, and she had several rotation students that were helping her. She was at the first year, she just started a lab, and she had this amazing finding that one gene that it's important for a developmental stage in this particular tiny and seemingly irrelevant organism, this worm, earthworm, actually these mutants were living longer than the wild type. That means that if you inactivate one particular receptor or you make it less active than the wild type receptors, the worms were living longer. So this is a survivorship curve. This is time in days after hatching. On the y-axis is you have the percentage of individuals that are alive. The white are the normal worms. The black are the worms that have like an inactive form of one particular type of receptor. So what this result showed was that knocking down a gene in the worm, it's sufficient to extend the lifespan of the worm in captivity. Worms were also more active for longer. They were reproducing apparently well. The worms were doing better. So this was really a turning point in the field, because then it raised the question, okay, but then are there genes that otherwise limit lifespan? Is there something like programmed to keep you less long-lived or aging? Is there a program for aging? So this was a big deal in the time. It was kind of like an expected result. And really, this is a seminal paper to many of us, and really she's been the pioneer of this whole field. And one little thing maybe for the student is that she couldn't convince any of her rotation students to be the first author on this paper. So she volunteered to be the first author on this paper, because all the rotation students that she had in her lab would not join her lab, because she was this new kid on the block and nobody wanted to stay with her. They would rather go with Highflick or with other more reputable kind of professors at UCSF. So she eventually she was the only permanent lab member on this paper. All the others were kind of rotation students that gracefully agreed to be on the paper. But this is most cited paper by far of the whole field. Okay, this raised a lot of questions about the evolutionary relevance, the evolutionary implications of why we age, how evolution has dealt with aging, etc., etc. And let me tell you this, that since this, seminal work that has shed a lot of light on the molecular mechanisms of aging in a worm, in yeast, budding yeast, these are kind of like baker's yeast, the ones that are used for beer, bread, etc. And in worms and in flies, we've found thousands, not in my group, but the community has found thousands of mutations that lead to extended lifespan, okay? So there is a very rich literature that shows that knocking down, and overexpressing certain genes in laboratory condition leads to extension of lifespan in these organisms, okay? And improving also response to stressors, external stressors. And these have to do with different molecular functions, like insulin IGF signaling, mitochondrial function, dietary restriction, mimetics, germline signaling, proteostasis, autophagy, stress response, and many more. I won't go into any of this detail because I want to share with you more of our work. But I wanted to show you that there's been really a revolution since the early 90s in the way we think about biology of aging from a genetic standpoint. Is there a question? These are knocked down, actually. This is inactivated, yeah. Is actually a genetic mutant that actually the receptor is less active in transmitting the signal when it binds insulin and the other ligands. Well, for IGF-1, many of these are actually knockouts and overexpressing, of these thousands. So the knockdown, the lower expression, the lower activities for the DAF2, the seminal paper, the first paper, but there are multiple knockout and knockdowns. Okay, so this begs the question again of, so what's going on with natural selection? Is natural selection favoring short life and active aging so that when you knock out those genes, the circuitry that controls that, then you live longer? What's going on there? This is been a very critical question. Well, that question might actually might be legitimate at first, but actually if we think a little bit deeper about how evolution really acts, then actually our perspective can shift properly, in a proper way, probably. So this is a typical way in which evolutionary biologists think about, when we think about, Darwinian selection, evolution, and selection. So you have a multidimensional space of genetic variants. So you have sequence space, genetic sequences, and then you have this sort of fitness peaks. So it's like you have this rugged landscape where if you start from a given point, like a genetic makeup of a species, then you have a series of mutations that bring you away from that point because the genetic distance increases from your starting point. And then you might actually reach this local maximum here, this little hilltop. And this hilltop maybe makes you more fertile, more fit from a selection, from a Darwinian standpoint. You produce more offspring than the valley where you were. But even there might be other paths actually that might lead you, maybe longer path that might lead you to higher peaks. Right, and this might take a lot of mutation, very extreme mutations that make you more fit, more able to transmit your alleles to the next generation. Actually, those same alleles might actually favor your fitness long term. What about aging? What about longevity? Well, longevity and aging actually might be completely orthogonal to all of this, because what we're talking about really, when we think about Darwinian fitness and what selection really cares for, evolution really caring for, is actually the reproductive output, right? So you might be living very long, but actually your reproductive output might still be the same as your starting point. And those variants that might be making you longer lived actually might not be selected for. Actually, you might even creating issues at selection point. So we have to a little bit reframe the way we think about possible selection for extended longevity. Of course, if you can live longer and reproduce longer, then of course it's substrate for selection to act upon. This is undeniable, of course. But for most of those variants, that's not the case. Okay, is longer life negligible aging promoted in evolution? And I want to indulge for this one slide, and then we get into our own results next. So there are two ways to think about how mutations, genetic variants that actually cause aging, that contribute to the aging process, that might be limiting longevity, come up. And these two frameworks have actually recognizable names in biology, in evolutionary biology of aging. but also they also reflect two different philosophical stands to the way we think about evolutionary biology in general. So let me tell you about them a little bit. So when we think about natural selection, for example, we think about DNA, so a bunch of ACGTs, and then you have this red C up there, and then this. So this variant is a new germline variant that comes, for example, in a new generation. And this variant, by pure chance, this is a complete exercise in thinking, like a thought experiment, if you wish. So increases fitness, so increases resilience, homeostasis, if you wish, and increases also higher, and it also leads to higher reproductive success. So on all accounts, is that provides higher fitness. So the individuals carrying those variants will be very successful, and it's the odds for that same variant mutation to be more frequent in the population in the next generation increases with time. So normally this type of if you sequence a bunch of individuals even in this room, you could see that compared to the ancestral population that did not have that red C but maybe had a G, a T, or an A there. Now, soon enough, if this variant is actually good, actually this might become very frequent. The frequency of this variant will increase. It turns out that this variant also leads late in life to age-related diseases, but that's really doesn't really matter to selection, because like I said, on the left side, the algebraic contribution of this variant to fitness is actually positive. So you have actually a contribution to fitness. Even though we might not like to become demented or slow or whatever, late in life, actually that same variant, actually it's selected for, okay? So this is one way we can think about how these aging variants might evolve. Okay, there is another scenario. Okay, yeah, this little diagram here shows that the frequency of this variant, this C, is 0 in the population just when it comes about, and then it becomes more and more frequent with time. This is a typical way of kind of like selective sweep by positive selection. This is how in jargon this is indicated. So let's put another example to the right. This time we have a red A, new germline variant, has no major impact on fitness, it's like seemingly neutral. However, late in life is actually bad for you because it makes you inflamed, cognitive impairment, and so on. Now, the statistical genetics of this type of latter variant is completely different from the previous. You won't actually increase the frequency of that 1A, but actually we'll have several new variants, a few new variants, all different from one another. So actually, if you take a group of individuals, you can know which variant actually have evolved neutrally, like here on the right, or more have been selected for. Either one of these variants actually does contribute to aging. You see, it's actually age-related diseases. One has been selected for because it actually turns out to be so advantageous for other reasons. This one has actually not been cared for by natural selection and has spread at lower frequency in the population. So these two kind of perspectives actually reflect two great, a big divide in the field. It's antagonistic pleiotropism. Don't forget, actually, it requires evolution to be actually selection to act. Positive selection, adaptive evolution. This was carried through by Williams. And mutation accumulation, Haldane, Meadawar, et al. So this is more kind of like a statistical genetics, but it's actually nearly neutral evolution is what really interpret this kind of variants. So let's go now to the work that we do. So we try to understand exactly what I showed you before and how much it matters in the context of aging in real populations and species. So we study aging at two levels. At the ontogenetic level to the left, so we try to understand and describe mechanisms of aging in organisms. We try to understand the molecular physiological basis for how and why organisms age, maybe try to intervene on those processes to improve health and homeostasis. But we also study the one part on the top, which is actually the evolutionary origin of those variants that actually cause in extant populations to age faster, slower, or to have a particular organ that ages. So we try to identify the evolutionary origins of variants, genetic variants that contribute to the aging process. One way that we like to think about, evolution is kind of like this tree structure, right? So you have the past to the top, and then you have these bifurcations, right? So it's a dichotomic tree. So every time you actually bifurcate, and this indicates actually the origin of new populations of new species. Let's say that we have this one red dot at some point in the past that actually leads to extension of lives. So this is a genetic variant that leads to a slight extension in lifespan in the individuals carrying it. So you have in this branch like a new variant that emerged, and this one branch is shared by these two species, and so both species will carry it in the present. And actually, maybe that variant leads to shorter life. Maybe there is like a new variant in this background that even leads to shortened lifespan and so forth. So we can try to historically understand and trace back when these variants emerged and how they actually have impacted longevity and aging in extant populations. And our favorite system, as indicated before by Tommaso Pizzarusso, is actually the African killifishes in particular, killifish. Right, so this is a fantastic clade of fish, and as biologists, we are really fond of our model system. We like to spend time with them, and we are really amazed and enjoy actually observing them. So there is a factor of fascination for the species per se. Oftentimes we are hobbyists, and we actually like to to take actually bugs in the fields, etc. But this research in the killifish has really helped us understand the genetics of aging, we think also beyond the killifish as a species, and I hope I will get the time to get there. So turquoise killifish in particular are the shortest known lived species among all of these African killifish species, and as mentioned before, this is something we started working on a long time ago with Sandro, with Eva, Tyrone, and others. And so this was a really fun time, and so these killifish are characterized by a natural short lifespan of a few months, 4 to 6 months, up to 8 months, depending on the strain. They have a quite dramatic age-dependent transformation. They undergo this spontaneous organismal and cellular and molecular aging transformation. And I will tell you a little bit of these changes. I will share some with you to convince you, I hope, that actually what aging means in this species has something to do also with the way that we age. Well, here I can show you, for example, that I recognize a cataract in the eye of this fish, of this male fish here. The fin pigmentation pattern in the tail actually starting declining. This is the full adult individuals. These are reproductively active. This female is starting to having a hunched back. So you see like this slight transformation, which are subtle, but actually we can do better and measure things, right? So we can do CAT scans, and we can actually see that bone density decreases spontaneously as a function of age. Actually, killifish develop spontaneously neoplasias, like these melanomas in the tail of the fish. An adult fish has a normal yellow-black stripe, but oftentimes in longer-lived yellow-tailed strain, you have the spontaneous onset of these pigment aberrations, which actually we're studying in our lab. And also many groups are interested in the brain of the fish, of the killifish, because it undergoes dramatic, spontaneous transformation. For example, here you have coronal sections of a young and an aged fish. This is a paper published by colleagues in Belgium, and they saw actually that senescent-associated beta-galactosidase, which is a marker of senescent cells in the brain, increases spontaneously and dramatically as a function of age. This is a cellular marker, a recognized and agreed upon marker of aging at cellular level. Okay, so these are just a few of those changes, but actually you can do more with the fish. You can also study integrated phenotypes, for example, learning, behavior. Fish actually have complex behaviors. You can actually train them to learn an association between a conditioned stimulus, a light, and a non-conditioned stimulus that they hate, air bubbles, in the tank. And they learn that every time there is light, that it's bad news because there will be bubbles, and they hate those bubbles, so they learn to avoid the bubbles by moving to the dark compartment before the bubbles come up. You can keep training them for 50 consecutive trials, and you can measure the success rate at every single trial, and then you can have a sliding window of success. And what we observe is that young adult individuals have a performance which is actually much higher than the old individuals. The old individuals learn, however, the performance of the young individual is higher. The first time point here is not the first trial, but is the average of the first 10 trials. So there is this kind of like learning deficit, if you wish, as a function of age, at least as measured by this active avoidance task. This is very useful because then we can make interventions, drug interventions, genetic interventions, and we can study whether the fish learning has been affected as a function of age. And this is just a very brief plug to a story which is now in preprint, and I will just summarize briefly. What you see here is actually the performance over trials, 40 consecutive trials. This is work done by Dennis De Backer, a phenomenal postdoc in my team. Dennis actually has knocked out a gene, APPA, amyloid precursor protein A, And he has observed that fish that lack amyloid precursor protein A actually learn better when they're old, when they're 7.5 months old. This is a long-lived killifish strain. They have better learning performance than the age-matched control fish. Not only that, but Dennis looked into the brain of this fish and looked at actually neurons that get activated with this early CFOS, this immediate early genes, and this kind of like this purple, this magenta indicate actually, so the blue are actually nuclei. This is a section of the brain of the fish. And the blue dots indicate the nuclei in APPA knockout, and here is the wild type. And actually what we observe is that early gene expression is higher in after learning in fish that are knockout for this gene. Long story short, we see that there is a sort of like a improvement of learning performance and brain activity in fish that lack this particular gene. So we are not affecting lifespan here, going back to the question maybe before, but with a knockout, we can actually increase learning performance. And actually the proteome, I won't go into detail of this, but what we see is that there is a proteome shift. So there is a proteome profile that changes as a function of age in the brain of the fish. And the knockout fish, when they're old, they resemble more the younger fish proteome. So there is also, in a way, a improvement in a delay in aging of the proteome of the fish. Okay, I want to also mention one other recent result by Silvia Cattelan, a phenomenal postdoc in the lab. She's so Dennis is more of kind of like histology guy, is a molecular biologist. Silvia is more she's a evolutionary ecologist, so she has a very different angle to things. So Silvia is an expert in actually male reproductive aging. And this is a picture that we took 2 years ago in Zimbabwe during our field activity. We were caught by a cyclone in the field. I will probably show you a few slides in that. So this is just to show her stubbornness and and resourcefulness. Okay, so Silvia studies actually reproductive aging in killifish, and what she observed is that actually sperm viability declines dramatically with age. This is measured by using dyes that enter the cells that are broken and then color different, and then different colors indicate whether the cell is viable, the sperm cell is viable or not. So she extracts ejaculates from individual fish, male fish, and then she measures these features of sperm activity, viability, and fertilization. Also, the DNA integrity dramatically drops between adult and old fish. And also, there is a little video here. You can see actually the individual sperm cells that move around. So you can measure actually sperm activity, velocity, and how many are active. And what Silvia discovered, and this is really like, she's amazing. She came with all this kind of knowledge, also methodological, about how to measure these things I had no idea about. So she discovered that motility goes down with age. Motility means how many cells are actually active. And velocity doesn't change. So of the cells that are actually moving, velocity doesn't change between young and old. So old fish have fewer active, alive cells. Okay. So, and fertilization success also, as a function of all of this, perhaps is impacted. She has also done longitudinal studies, not cross-sectional, like I just showed you, at different ages, and she sees that actually the embryo survivorship changes as a function of father age. So again, she does in vitro fertilization, and from the ejaculate, she actually can observe from the same individuals that the fertilization rate of individual 1 at age young, middle age, and low and old actually declines dramatically. So across all these individuals. And even more strikingly, this is unpublished work, what Silvia observed, again, longitudinally, is that offspring of young fathers live longer than offspring of the same fathers when the same fathers are old. So she extracts sperm from young fathers and the same fathers when they're old, and then she generates this new generation of offspring, and then she measures survivorship in offspring from the same fathers, as young as old, and there is this difference. What about females, you may ask? Females are all young adult females. So we don't have young mating with young and old mating with old. Young, so the age of the female, we can factor it out. Okay, let's go back to killifish and the genetic of killifish. So this was just to show you how dramatic and yet undiscovered, uncharacterized aging in killifish is across different systems, across different organs. And I'm really like talking very superficially because there is, of course, much more results that I'm happy also to tell you about later if you want. So killifish are a very diverse clade of teleosts. They occupy different biotypes, different habitats in Africa, dry and wet. And they are a particular type of adaptations. But what's striking and what really helps our case to identify the genetic basis of lifespan and longevity is that spontaneously you have short-lived and long-lived killifish, even within the same strain. I already mentioned that before. Dennis, the postdoc, was studying long-lived killifish. But we also have within the same species long-lived and short-lived. This is very useful because you can do genetic crosses, and then you can study the segregation of the trait longevity in a cross. So you can see how longevity is inherited as a phenotype in the F1 and the F2 generation. And then using genetics, quantitative genetics, you can actually identify regions of the genome that correlate with being long-lived or short-lived. They are simply associated with them, right? Yes, it's heritable. Indeed, that's what the next slide shows. That depending on the genotype at one particular locus in this linkage group 3, you can actually predict lifespan. So the genotype at this particular marker 46347 in the F2 generation of this cross predicts how long-lived you are. So you can have both alleles from the long-lived grandparent and you're long-lived. You have just one allele from the long-lived grandparent, you're also long-lived. If you have both alleles from the short-lived grandparent, you're short-lived. This is not maternally inherited, it's not mitochondria, it's really nuclear genome. And so this really is the substrate for our studies on the genetics of aging. So we can actually see. So actually, so we cross the two F0, so different directions of the cross, and then we measure survivorship in the F1 and F2. But the parents, actually, we use the mean phenotype for the parent strain. So we're talking about different strain, which have a median phenotype of 4 months for the short-lived and 9 months, for example, for the long-lived. And then we study in the F2 the segregation of the trait. And so we can tell apart the longer-lived or the shorter-lived, what type of alleles do they share among themselves. These are all cousins, right, from the same grandparents. And so we can enrich we can zoom into regions of the genome associated with longevity. And then we can go into those regions of the genome and we can look at what genes, what genetic variants are causing longevity in them. However, this is for anyone who actually knows genetic mapping and quantitative genetics. This is very interesting. This is very exciting. We published it. We were very excited about it. However, this is a nightmare when it comes to genetics because this chromosome 3 is a sex chromosome. And so it has a huge region that actually is non-recombining. That means that if you do genetic mapping, you need recombination to break down the region. And actually, this is the very one region of the genome that is completely devoid of recombination. And this is what prompted us to actually look more broadly into the genetics of aging from a population genetic perspective, from a more statistical genetic perspective, rather than crosses. And that's what I hope I will be able to do in the next 2 minutes. Right, so the genetic of aging and of lifespan is complex. It's not simple. It's not 1-2 regions of the genome. It's actually polygenic, it's extremely polygenic, even though there are regions that have a large explained variance, but it's actually extremely polygenic. That's why we need to go and look at the wild species and the wild populations and their variation in lifespan and aging phenotypes. So this is what we did. We went already when I was a PhD student with Sandro. We had the fortune to actually go. It was amazing to be a PhD student and go in Mozambique to find killifish in the wild. And we brought them back to the lab. And a lot of labs in the world now work on those one strains that we collected back in the day. So there was a great time, great fun. And we kept going back to the same region. So we have now a scientific station in Zimbabwe where we do field work yearly. Actually, last month I came back from our field research activity. Killifish live in a very striking environment characterized by rainfalls and dry seasons. So they are adapted to an environment which is very predictable in the sense that there is only rain for a few months every year, but most of the year there is no water, no rain, and the embryos live in the dry mud like here. So we go to the field to do the field work. This is just a funny slide to show you our encounters with elephants. It turns out that actually this slide actually has a sense because actually killifish share their environment with elephants. Indeed, these are actually water holes where elephants go and do their business. And this was actually March 2023. Sylvia, I showed her work to you before, and this is Sam, a PhD student in the lab. They're trying hard to collect killifish with this net in the pond. And this is actually last year in February. I tried to take a picture from exactly the same region, so I was just making sure I was outside of the pond and while they were doing the hard work here. And these are actually elephant tracks here. So anyway, this is just to show you how complex the environment where they live in are, and just a couple of slides of the environment where we work. This is a typical killifish pond, and this is how we process the samples in the field. This is a bottle of ethanol. These are gloves. Sylvia is labeling and dissecting gonads in this fish, and here we have tubes where we collect our samples. Then we ship back to Europe for analysis. We are fortunate to work with the Gonada Zoo National Park. This is the place where these killifish are, and this is just another session of dissecting fish in the field. Okay, so these are how the fish look. What did we discover regarding the genetics of longevity? Well, we found that actually I will just summarize a lot of years of work that was published some time ago. What we found is that actually the evolution towards short lifespan is actually not adaptive. This is what we see. So there is no adaptive evolution towards shorter lifespan. This is our kind of corollary understanding of this whole activity. And I want to start with this statement. And most of this work were done by two phenomenal lab members. I mean, Ray and David Williamson. So Ray discovered that the genome of the fish, the genome size of the killifish, co-varies with whether they are annual, whether they live in these extreme environments, or whether they live in environments that are normal, like, water environments like lake and rivers where non-annual fish live. In particular, short-lived killifish, annual species of larger genomes sizes, and this genome size expansion is actually driven by proliferation of transposable elements and repetitive regions. If you mask out the repetitive portion of the genome, then the size of the genome of the annual fish is the same as the size of the genome of the non-annual fish. So we can rank actually the contribution of each of those transposable element and selfish elements in the genome from the ones that contribute the most to genome size expansion to the ones that contribute the least. Interestingly, this is not only a macroevolutionary phenomenon, so the fact that genome size co-varies with whether a fish lives in a dry environment or a wet environment, but also it's a microevolutionary feature. We discovered that even within a species, for example, the Notobranchius ortonotus, Notobranchius rakovi, if you are a population that lives in a wet environment or in a dry environment, wet and dry, your genome size will be larger if you're in the dry environment and smaller if you're in a wet environment. There is a genome size expansion in the drier environment, and we believe this is not adaptive. This is simply the consequence of mutation accumulation and uncontrolled genome maintenance in this species. And I hope I will have the time and the mean to convince you that that's the case. Please.
When you do these experiments, do you send always to the same pond, or you work in different ponds and compare what happens?
Both. So we do. Yes, these ones, for example, come from the same ponds. The wet and the wet live in the same ponds. The dry and the dried live in the same ponds. And so we see this kind of parallel species-specific change in this genome size, but different species though. Yeah, I will show you in a few slides. We did exactly that analysis. I will show you. So we have all these locations that we have sampled through time. So these are the yellow dots that we and others have collected, where we have collected fish over time. This is a climate map of Africa, of this part of Africa. Red means dry and hot, short rainy season. Blue means wet and colder, longer rainy season and multiple cyclones per year. So what we observe is that actually in the wet environments, you have large ponds. You have a longer living ponds. You have larger populations. In the dry environments uphill, where the short-lived fish come from, you have fragmented populations. So there is gene flow here, oops, and there is very weak gene flow here. There is gene flow downstream from the dry ponds to the wet ponds because it goes downstream, the water. But actually, there is population fragmentation. And in statistical genetics, we know that actually selection is very weak when genetic diversity, selection is very weak when genetic diversity is low, when effective population size is small. this is what we measured here. So this is the work of David Willemsen that was published a few years back. So we see again, this, to go back to this question, we have large effective population size in the coastal region, smaller population size in the inland region, and smaller population size even inside. This concept, population size, is in bracket. It's in means genetic diversity. This is effective population size. It's a measure of genetic diversity, okay, which is the substrate on which selection acts. If you have very limited genetic diversity to play with, selection cannot really favor the most advantageous alleles. It's like when you play cards. If you're handed like a small, a few cards, or if you have a lot of cards, but they're all just the same, you can't do much, you can't win, right? So this is exactly the same thing here. We see that reduction in population size. And this is how you measure reduction in population size. You sequence a lot of fish, and then you look at the linkage disequilibrium and the fragmentation of and random homozygosity, doesn't matter. And then you see that historically population size crashed. So this is the present, and this is the past, and this is population size on the y-axis. And actually you see that this one population, which is the dry one, underwent expansion and contraction. It's very small population size. The longer lived, the wet population size, the black, the gray, and the blue, actually underwent recent population size expansion. So we see actually with our eyes that selection is supposed to be stronger to purge the genome from deleterious variants in larger, longer lived populations. This is another statistical measure, and I promise I won't go into that. All that this indicates is that indeed we can measure the strength of selection, of positive selection, and the strength of selection, so the capacity that natural selection has to remove deleterious variants and to favor beneficial germline variants is actually weaker in the short-lived fish. So this is actually the McDonald-Kreitman alpha asymptotic. It indicates exactly that, the strength of selection. The lower red curve indicates that the dry populations have a weaker genome-wide selection using different controls of outgroups. And if you look at the genome and you ask what does it really mean, it means that the short-lived populations actually have more pseudogenization, more genes, and the vast majority of the genome has undergone, compared to the other populations, missense variants, stop gained. So the color of these bars indicates different type of genetic event that are unique to each population. And the same is true also for other species, not just the turquoise killifish, but also this spotted killifish and the Notobranchius rakovi. The populations living in dry environment underwent continuous population bottleneck, really exploding the mutation load genome-wide. We also have studied, and the same for the McDonald-Kreitman test, we also have studied the mechanistic impact of this genetic catastrophe in these kind of bottlenecked populations. And but I won't explain that to you now, but this is published work. So to summarize, we have population size decline that leads to declining efficacy of selection in parallel to population size. And that leads to deleterious genetic variants increasing genome-wide in those shorter-lived populations, in those populations that have a smaller effective population size, those that also have the larger genome, you remember. And that actually impacts, we believe, lifespan in these species. These populations don't really vary for how fast they reach sexual maturity, at least not dramatically. So there is no real correlation between how fast they are in reaching sexual maturity and how long they live. So these things are separated. But there might be more with embryonic phenotypes, which are actually very interesting and on which Sandro's lab is working. So going back to this fitness landscape, if you think that you have all the freedom, your genetic space, your space for choices of genetic variants is infinite, then you can climb any mountain, any fitness mountain. But actually, it turns out that the short-lived, the populations which are constraining those, uphill, isolated populations actually have very limited genetic diversity to play with. And so they have very limited opportunities to actually climb any fitness peak. If anything, actually, they sink down because any new mutation will be much more likely to be deleterious than beneficial. So this is kind of recipe for disaster from a genomic standpoint and from a fitness standpoint. And we believe that some of these populations are at the brink of extinction. What comes to the rescue is actually hybridization. So we think the hybridization really comes to the rescue from time to time. Those major climate events come to the rescue. This is a summary slide. we don't deny that there is very strong positive selection in these populations. Of course, living in this harsh environment leads to selection to find solution to survive there. We think that most of those solutions have to do with early life, though. Embryonic diapause, surviving in the dry mud for a long time, and having an explosive sexual maturation. Maybe some of those genes selected, and we see them. We find genes that are under strong selection: thyroid-stimulating hormone, vitellogenin, luteic hormone, and many others involved in early life phenotypes are under very strong selection, even in the very short-lived species. So selection is, of course, strong and working there. But for the vast majority of the late life acting genes, the ones that are non-embryonic, mostly developed, expressed in adulthood, many immune genes, relaxation or purifying selection leads to a catastrophe: genome-wide genome size expansion, proliferation of transposable elements that, of course, can also be the substrate for adaptive evolution, but it's a lot of stochasticity there. It's a big risk to run. And also, I didn't tell you about that, mitochondrial genome expansion. Okay, I don't think I have much time left. So I will also tell you that the results that we find, so we see that population size impacts lifespan and the genetics of aging. And I wanted to say that it might be that actually lifespan and population size might have like a unobserved confounder. So we see this correlation. We see some actually causal link there, but there might be some actually shared confounder up there. And so we test this also in using simulations and forward simulation, and then we can generalize our findings. And this is actually published as a preprint. So if you're curious about to know more about this, I'm happy to tell you more about this. But yes, we do see that in general, population size actually impacts directly lifespan under both mutation accumulation and antagonistic pleiotropy. And I'm happy to tell you more about this if you want offline. So to maybe conclude, what we think we are seeing here, it's some quite fundamental review of the way we understand the biology and the genetics and the genomics of aging and the evolution of aging. We're sort of like we focus our attention on the mutation accumulation in the context of population size. And so we really raise attention towards demography, pastdemographic events, the ecology that this actually shapes demography and ultimately shapes the distribution of these variants genome-wide and their impact. And just as a concluding slide, we see that antagonistic pleiotropy, so this selection-based aging, is still possible, but the prerequisite for that to happen is that population size has to be large. Otherwise, selection just doesn't work. And in this case, you have most likely, at least in our model, antagonistic pleiotropism. In the context of small population size, limited genetic diversity, you have this sort of like increase of deleterious mutations genome-wide, and these deleterious mutations stick around in the gene pool without being purged, without being weeded out by natural selection, because simply selection cannot weed them out. By the way, the later these variants are expressed in life, the later these genes carrying these variants are expressed, the weaker, even more, is selection in purging them off. So even in large populations, genes that are expressed preferentially in late life or rarely expressed actually are also, they themselves have a small population size if you think about it, because few individuals express them in the population. Okay, so I read somewhere that nothing in evolutionary biology of aging makes sense except in the light of demography. And we think that this also matters to us, to humans. We think that we should really worry about this or be concerned or aware of this phenomenon that we observe, because we, as a species, have a very small population size. You may say, oh, but come on, we are 8 billion. Well, that's the census population size. Our genetic diversity is extremely low. It actually counts down to about 40,000 individuals. 40,000 is the effective population size accounted for the human species. Mice, in comparison, wild mice have a larger population size. What does that mean is that our background for variants to be selected positively or negatively is kind of like weak. And in particular, this is a big problem for out of Africa populations. So you have this kind of fronts of migrations that have selected, that have drifted variants in a very private manner, and this actually correlates with the population-specific aging patterns. So we think that our findings also matter more broadly in the context of human population history and population genetics. With that, I'm done, and I would like to thank my lab, and I told you a little bit about the work of Dennis here, and of Sylvia here, and of David here, and of Ray a little bit also. I didn't tell you other stories related to the microbiome and immune system. I hope there will be another occasion, but also I'm really thankful to all the organizers, or all the collaborators, including Sandro Cellerino, with whom I have great ongoing collaborations. And it's always a great fun to discuss science and to think about the next great ideas. And also I'm thankful to our funding sources. And I go back, and if there is time for questions, I'm happy to take them and hopefully answer them. Thanks.
That was super interesting. However, you mentioned several times the word spontaneous when it comes to aging, which is something I don't understand. Like, for example, in female women, menopause has been linked to accelerated aging. So it seems as if there is a mechanism that somehow, for some reason that perhaps we don't entirely understand, is put into place and activated at some point in the person's life. And so aging is brought about. But why spontaneous in this regard?
Okay, thank you for your question. So your point is about spontaneous versus programmed. Is that what you're getting to?
It should be called spontaneous. That's what I don't understand, because it seems as if it's something systematic that somehow is activated at specific point in time.
Okay, thank you. Yeah, so the way I use spontaneous is to indicate something that is not triggered by external stimulation, for example. So it occurs uninduced. So for example, if you study neurodegeneration in mice, if you try, you have to sort of really push hard. You have to create, multiple knockouts to generate the phenotype you're in. So in that case, it's not spontaneous. It's like heavily induced. In killifish, for example, if you want to study neurodegeneration, you just wait, because you know that actually if the fish is living long enough and you observe and you have your patient, then the neurodegeneration phenotype or the brain aging phenotype will occur spontaneously, without being induced. But maybe mine is a misuse of the term spontaneous, because in Italian, I'm Italian, so maybe I'm thinking spontaneous with a slightly different connotation from the English use of the word. And I would appreciate if you tell me that's not how spontaneous it's used.
No, that's fine. Perhaps from the point of view of ecology, it is spontaneous, but if you go to the hormonal level or physiological, it's really perhaps something else.
It's induced by a chain of events, which is not depending on external factors. It's probably like set emotions, but we don't think there is a program for it. If that's what you mean. It's like shelf life in a way, right? So if I have a loaf of bread which has a shelf life of, I don't know, a month, after 1 month it will decay, will mold probably, right? But it is not that, the bakers have engineered the loaf of bread to mold, right? It's just happening. That's how we see it, right?
Great, thank you.
Sure.
Hey, so I have 2 questions, maybe 3. When you said that in your experience, a short-living strain and long-living strain, they reach sexual maturity more or less at the same time. Allora, how more or less is this more or less? Because, I agree. But if I think that they reach sexual maturity at 4 weeks and that my eye cannot distinguish 1 week difference, 1 week on 4 weeks is 25%. And this strain live more or less 30% more than the other. So do you have any quantification of these? They reach the same, or is just...
Yeah, of course. I wouldn't have said it otherwise. It's not a gut feeling, it's a measure. So we measure onset of sexual maturity by fertilizing eggs. So Sylvia and Dennis, so I had exactly your point to Sylvia and Dennis. So they measure sexual maturity in Fritz, different strains, and Cadlitzi, different, and Nosobranchus Cadlitzi. At least for those 2, species, sexual maturity onset, measured fertilization, when you start fertilizing embryos naturally, but also with IVF, it's actually indistinguishable. Yeah, you're right. Maybe there is a few days difference that still might make a difference in nature if they coexist, if they co-occur. But if anything, the shorter lived seem to actually be a little bit later. But
So it was assessed by fertilization, not by tail coloring or whatever.
Yeah, of course. Yeah. No, color is, no, it's dangerous. Yeah, exactly.
No, yeah. Then the other thing, the other thing is, so the short-living strain. This is a
unfinished debate. Yes, I know what you're asking.
Strain. Is aging faster and for this living shorter, or is kind of aging more or less the same, and at some point it dies because of this overriding alteration, mutation, blah blah blah?
We see that the very shortest lived strain, which is also the most inbred line that we have, right? So this has been collected in 1969. It's been ever since inbred. That means that also has been, adapted to laboratory conditions. Is an artificial system. And as Sandro's work in particular has shown, but also we see the same thing. They start off as aged, if you wish, from a, omics perspective, right? So they start off as if they were older already, right? But also there is actually, so the slope of aging might actually not really change compared to the longer lived strains. They might actually be parallel in the way they age, but the shorter lived seem to start off already quite compromised. Sorry, I can't give you much more than this, but Dennis is trying to address exactly this question by doing a QTL mapping for brain aging using short-lived strain and longer-lived strain. But maybe we can take this conversation afterwards, Luca, because I'm not sure if everybody, but yeah.
The third one was about methodological, about the sperm. If you can go to the slide.
Silvia's slide, right?
See. The male sperm of different age fertilizing eggs. See, more or less. No, yeah, more or less. So you
this wait.
See, this was done. This was done with fresh sperm or frozen sperm.
Oh, fresh.
Fresh. And how do you avoid the batch effect? Because if you fertilize with fresh sperm from the same individual, female of the same age, this female have to be different.
Of course.
So how do you minimize the batch effect?
Yeah, you stagger your cohort.
Okay.
Of males. So you have young males, the same females, and you have young males and old males, and then you have other females, young male, and
okay,
So, I also have a very broad question. Of course, I would be curious to know about the microbiota part, but we can talk about that later. But did you? Of course, I don't think we're gonna live, 200 years as human beings. It's just a very broad question. But what we really care for is quality of life. So, do you think that they are? Did you ever seen an uncoupling between like performance in behavioral tests, motor performance, and longevity?
Yeah.
Do they think they really go together, or you can live a normal life? You die at a normal age, but you live better. That's.
I can give you just. I can speak with what I see from the fish, right? So, and in this work done by Dennis. On the amyloid precursor protein. So we see, by all means, signs of better performing brain when it comes to cognition, well, at least measured with that assay. And when we measure brain function with this way that we do that, with the limitations and the caveats connected to it, but these guys don't seem to be living longer, for example, right? So there I see a straightforward uncoupling. So this is what I can tell you. But this is really like brain. So this was tailored to the brain. And if you can imagine that maybe if this protein is, exquisitely expressed in the brain in one organ, of course you can have a cross systems benefit, but we don't necessarily see that. I can see ways in which indirectly you can also impact systemic health. But in these guys, we don't see, as far as I know, last I checked with Dennis, we don't see them living longer.
So you have, at least
Uncoupling.
for the brain, you have
Yes.
uncoupling.
Yes.
Maybe I have a comment exactly on this, and Dario also knows this paper, Urialon papers. So very recent paper, Nature Communication. They made a mathematical model, so a stochastic differential equation, where they basically model just what Dario said. The production of damage, so they model a damage process and a repair process that, however, work differently. No, the repair process is like sigmoidal and then saturates at some point. Now, in that model, if you just improve the repair, you don't prolong the lifespan, but the curves become more and more rectangular. So that would exactly say that if you work specifically on the repair, you compress the part of life where you have disease, which is basically the aim of our entire field. So to compress that part of life.
Comorbidity, compressed comorbidity.
And Dario, I have a very general question. I think it's very important to, convey the general concept that our population size is very, very small.
Yeah.
Because not many people are aware of this. However, we are also very, very long-lived. And
it's true. It's not only true for humans. Also, whales are extremely long-lived, very small effective population size. Some rockfish are very small population size, extremely long-lived. Yeah, it's not true only for humans.
And so, basically, based on your model, you would predict that, like African populations that have larger population size should have slower aging.
In principle, with the shared equality world, maybe that would be the case. I think socioeconomical status, it's a big confounder there. So yeah, I think it's an important point. And I think there is no such study. We're actually looking now into African centenarians also. This is something I'm also interested, because I think that there is a unmined, untapped resource in terms of genetics. It's wonderful what could be found there, could be explored with colleagues in Kenya. And but yeah, the model is actually exquisitely microevolutionary model. It's not across species, right? So we can't make leaps from species 1 to species 2, and then species 2 is longer lived than species A because has a larger population size. It's really within a species. We let species evolve in our model, and we can predict really that genetic diversity provides a substrate for variants to be selected to enhance health. I mean, at the end of the day, it's kind of trivial what we come to see, but actually it's supported by the empirical evidence.
But the nice thing is that, we have sort of an unfortunate natural experiment. If we look at African Americans, they are bottleneck of the very recent bottleneck of the African population.
Yeah, I don't know much
Even
about
though conditions are very different. But it could be interesting.
Yeah. So one thing I have to say about species like us, humans, that have small effective population size is that we have compensatory mechanisms. While we have small population size, we build genetic diversity from the back door. So, for example, our immune system, as maybe the biologists know, but maybe the physicists, the mathematicians, the chemists don't know, we have a system of random number generator in our lymphocytes, right? Our lymphocytes explore a space of variation of antibodies which cannot be explained by our genetic makeup, right? So our lymphocytes run dice and then generate like an astronomically diverse, substrate for antibody production. And then these antibodies are like little hypotheses about how the world out there is. So even if you have small effective population size, even if you have small genetic diversity, immune system has found a way, adaptive immunity has found a way to actually rest and stay diverse in the face of otherwise homogeneity. And this is what we actually see also in the field. We study also now, we started studying rhinos. We have these extremely inbred rhino populations with an amazingly diverse immune system, which cannot be explained by the population diversity, but can be explained by their exquisitely sophisticated immune system. So, yeah.
Just one thing about the experiment before this slide regarding the behavior exactly. Did you also check the decay in motility? Because one thing that came to
Yes,
mind.
absolutely.
Okay, because that one
Yes,
was.
absolutely. So we do that. There is some correlation, but it doesn't explain it all. So, as of course, if you take a immobile, like a fish that cannot move, you cannot perform according to this test. But also, if you have an extremely hectic fish, extremely restless fish, also they will not learn the association. They will get stressed. So at the extreme, you completely you lose completely signal. On average, younger fish are more active. But actually, I have videos I want to show you, Silvia, maybe if you want, where a fish that actually has learned the task is a fish that stays in the middle. Actually, it's really funny to see them. They actually wait in the middle for where the light goes. And if the light goes here, they just go like this, and they just flip and then just move like just a little bit like that. Or maybe with the tail, they just go one thing there. And then the light goes off, and then they go in the middle again. So exploration in the beginning helps them familiarize with the environment, but in general, the performance is kind of like cannot be entirely explained by motility. Yeah.
Okay, I think we can close here, and thank you again for this amazing seminar and the question and answers.
Thanks.
Thank you.